So it's really my great pleasure to introduce Dr. Min Yu. She will give a talk in our seminar today. And she did her undergrad and master's degree back in China in Shandong Medical University and Peking University respectively. And then she came to States to pursue her PhD degree. And she did her PhD training in State University of New York at Stony Brook. And then after that, she went on for postdoc training in Mass General Hospital in 2007. And she did a very good job in her postdoc. After just 1 year, very impressively, she became a HHMI research fellow in 2008. And she published very multiple top-tier research articles, including one Nature and 2 Science papers, where she is the first author. And in those articles, she disclosed the dynamic epithelial and mesenchymal feature of those circulating tumor cells. And she also discovered the therapy resistance mechanisms and the functional plasticity of those CTCs. So without any further ado, I would introduce Dr. Yu to give us his talk today. Thank you.
Hello, can you hear me? Can you hear me on the back? Oh, okay. So thanks for the very nice introduction. Thanks for the invitation from Hyde and Humboldt. So it's really an honor for me to be here, and I have really enjoyed my visit here. I met some new friends and also old friends we haven't seen in many years. So today I'll really talk to you about circulating tumor cells. And then my lab is really interested in how cancer metastasis. We focus on breast cancer because that's mainly the main mortality reason is the tumor cells that grow in the distant organs. For example, in breast cancer, often this is simplified view of metastasis, how it happens. So rare cells can shed into circulation, and that's the time we call it circulating tumor cells. And those cells, among those, a subset of those will eventually grow metastasis in different organs. In breast cancer particularly, there is organotropism. That means metastasis very often happen in certain organs. For example, very highly in bone, in lung, and the brain, liver, and sometimes ovaries. And we are particularly interested in circulating tumor cells because one reason is because CTCs can be sampled by minimum invasive blood draw. So it becomes a good biopsy, so you can sample multiple times. And then not only it contains metastatic precursors, but also the cells can shed from metastatic tissues. So that becomes a good liquid biopsy method for metastatic settings. So also, because you can sample multiple times, so that can reflect individual tumors' current signatures. So that, for those reasons, we're particularly interested in understanding more details by CTCs. So, for example, we want to know what are the metastatic precursors in CTCs that whether we can identify early and we can either predict where the metastasis is going to happen or we can develop intervention that to stop metastasis early. And also we're interested in what are the heterogeneity of the cell CTCs in the blood and how that contributed to resistant mechanisms. So, however, study CTC is very challenging. So this is relatively amount of CTC you can get from patient relative to millions and billions normal blood cells in the blood. So this is really challenging, like finding a needle in a haystack. However, bioengineers love this problem. So in the last 10, 20 years, there is many advances from bioengineer effort, and has developed many different technology to help address this question. So basically try to identify those rare circulating tumor cells from patient. And then the main approach, just summarize of several popular CTC isolation technologies. And the main idea is to separate circulating tumor cells away from the normal blood cells. So this largely can be separated into like 2 parts. One is based on what do we know about cancer surface markers, kind of using that as a positive selection. So most commonly used are the EpiCam. So this is the commonly used in a cell search that's been FDA cleared. And then there's also different versions using the positive selection. The other camp is mainly using negative depletion. So basically, we know very little about those cells in the blood. We don't know how heterogeneous they are, what are the markers that uniformly express. We may miss some population-based positive selection. But there's a whole set of technology based on the depletion of the normal blood cells, because we don't know much about CTC, but we know a lot about normal blood cells. So based on this, you can use size-based separation. You can use also white blood cell antibody to deplete those white blood cells and red blood cells. There are also many technologies combine different formats together as a way to isolate CTC. I would say there's no golden standard about which one's the best method. It really depends on, there's pros and cons for each one of them. It really depends on whether you need a fixed cells or live cells, or what are the downstream application, whether you need high purity or you need a high efficiency. So that determines what technology you want to use. So in the last 10, 20 years, there's a large study using cell search-based technology have shown that patients who have high baseline CTC numbers has worse prognosis overall. So this is mainly just looking at CTC as a prognosis markers, as counting how many CTCs in the baseline and also during the disease treatment. However, we think there are a lot of things we can learn on CTC beyond just counting quantities. For example, whether we can know much more about CTC characteristics, whether we can use really the molecular property of CTC as a liquid biopsy, and also to understand what's the mechanism for metastasis. So the biggest challenge for this understanding deeper CTC is mainly the quantities that we're talking about in a typical tube of blood. In the metastatic case, in breast cancer, you have less than 10, 20 cells in general. And in some cases, you have more, like 100,000, but those are rare cases. So how do you utilize those to really understand CTC biology? That's really challenging. So today this is the outline. I'm going to talk to you about a published work. This is the last part of my postdoc work. At that time, I tried to really to see whether we can culture the CTC in vitro to expand those cells. And that has been published. And then I'm going to spend a majority of time talking about a new unpublished work, which is in revision right now. And to basically to understand, using those established CTC cultures, what we can understand about the mechanism of metastasis. And the last part, I just briefly touch about our effort to improve technology and method to isolate live CTC analysis, so to perform live CTC analysis. So in this one project, I really try to see whether we can expand the CTC in vitro. So we use a method basically based on negative depletion of white blood cells. We conjugated with antibody with magnetic beads to deplete the white blood cells and red blood cells, and then leave the tumor cells untouched. And then, so this was previously we used CTC chip. That's when in postdoc time. And then we also can use this in the Rosette. It's a commercial available technology. Also can deplete white blood cells. So with this, we finally have certain success. So this is one example of 3 CTC isolated from a luminal metastatic breast cancer patient. And over time, it really grew into millions of cells. So that gave us hope that it's possible to culture CTCs in vitro. However, it's still very challenging. At the end, among many blood draws, we actually have 6 patients that are successfully expanded. So those are the CTCs from 6 patients and expanded in vitro and has been maintained for many years now. And then surprisingly, they're all from a luminal type of breast cancer patient. That was really surprising because we thought the triple negative patient typically have more aggressive phenotype, it would be easier to culture. It could be this culture condition just favor the luminal type, or it could be some other unknown reasons we don't know. This is what we validated that they really have. So after weeks and months of culture, the contaminated white blood cell, red blood cell all died. You left it with pure tumor cells, and then we confirmed by cytokeratin staining and Ki67, and then they 5 out of 6 patients still have ER positivity in the nucleus. And then to definitely confirm this is really tumor cells, so we did a deep sequencing about 1,000 genes panels that are commonly mutated in cancer. So we found that this is a summary of a list of the known driver mutations, and then we found that we compared in the patient. Where was it? Here. Some patient we have the same tumor biopsy from the same patient. So we compared whether we can find the same mutation or not. So in the cases, we do see the same driver mutation, suggesting they are from the same tumor. However, interestingly, we find additional mutation that we identified in CTC lines, but not detected in the primary tumor biopsies. And including, we actually, among the 6 patients, we found 3 of them carry 3 different point mutation on the ligand binding region of the estrogen receptor. So when we found this, when we prepared the manuscript, several papers published showing exactly the same finding, that the hotspot mutation in the ER, in the estrogen receptor, that are commonly present in the metastasis of the patient who become resistant to aromatase inhibitor, but not in the primary tumor. So we found the CTCs that are shed from, those are from metastatic patients, so they are shed from metastasis, they also carry the same mutation. So the reason that we found additional mutation is because the tumor biopsy taken was several years before, so this is diagnosis, this is where the tumor biopsy was taken, and then this is the red line are the CTC lines we established. So you can see that the patient, there has been through multiple rounds of treatment from the initial biopsy versus the time we analyzed CTC. So this additional mutation maybe represent the potential mechanism for resistance. So to test this, now we have cells, we can actually do a drug screen to see whether that's the case. So we're able to, using CTC lines, to do a small panel drug screen to look at cell viability. So this is here showing the 4 replicates for each condition with the control and the increased amount of drugs, and either single drugs or drug combinations. So you can see that each patient line with different mutation, their behavior to the certain drug is different. And we further tested one line, BRX7, because interestingly, this patient had both acquired mutation of a PIK3CA and FGFR2. So since the primary tumor have never showed those mutations, this patient never been treated with any of the drugs. So we tested in vitro by treating with single drugs targeting PI3 kinase pathway or FGFR pathway with 2 different FGFR inhibitors. You can see single drug was able to suppress tumor cell growth, but if it combined together, it becomes very synergistic. So this is probably suggesting those cells are kind of dependent on those oncogenic pathways. This is in vitro, and we injected the cells in vivo and tested it with the treatment of vehicle, or single drug alone, or combination drug. You can see that with single drug, you do see tumor suppression, but with the combination of 2 drugs, it really significantly suppressed tumor takeoff. So this was published. I went really quickly. So this is really showing that the utility of CTCs if you have enough material. So that's the time when I started my lab at USC. So the first question, serial question we want to ask is, so whether the CTCs being cultured, they can still form metastasis. And if they do, in mice, if they do they form in mice in similar pattern to the patient? So if they do have this similar to the patient, do they have this, well-known organotropism for metastasis? And what are the molecular programs determining the organotropism? And can we identify the biomarkers or drivers that we can eventually use to predict and monitor additional patients to see whether we can predict where is the next metastasis? So with those serious questions in mind, we have a project led by a fantastic postdoc, Remy Klaus, in the lab. So what he did is he labeled the CTC with GFP luciferase to be able to help in vivo and in vitro imaging. And he injected it in the mouse heart by intracardiac injection directly into left ventricle. So the reason, because then the cells in the arterial blood will have kind of roughly equal ability to go to all the organs. And then we want to see whether those cells form metastasis in different organs in mice. So we can start to see that mice have metastasis in the brain, the lung, the bone, the ovaries, and in certain cases, multiple metastases. So because they have luciferase, we can, this is a summary of 4 of those lines. We can image within 10 minutes how good the injection. You will see the tumor cells disperse all over the body. And then within in a month, you start to see tumor signal in certain organs derive. But it's very dynamic. Sometimes we see signals goes up and go down. And then in about 6 months, we do see steady tumor growth in different organs. And that's time we quantified, we summarized where the metastasis happened in the mice by percentage. So this is the graph summarize 4 different patient-derived lines where the metastasis happen on percentage in mice. For example, in this one, you see a lot of them are formed in the lung, some in ovary, and some in the bone. And this also overall have more metastasis, give a little bit toward the bone. And then these two have very high metastatic potential. These 2 lines have a low metastatic potential. But we start to see the brain metastasis arise from these 2 lines, BRX42, and then in this one, 1 out of 20 has the brain metastasis. So how does this compare to the patient? So in collaboration with Aditya Bardia and Dan Jurek from MGH, because those patient lines still was derived from MGH. So we compare where is the metastasis happening in the patient. So this is roughly reflect the major organs that we see in the patient. And in BRX7, we see lung metastasis. 68, we see a lot of bone metastasis. And in BRX42 is interesting. We see the brain metastasis, ovary, and bone metastasis. And 50, we see bone metastasis. And what's really exciting is this patient, BRX42, developed brain metastasis 1 year after we isolated CTC from her. So that means the cells we isolated could be metastatic precursors. So this is the first case we know of that we identified from the blood, the metastatic precursors, that later being confirmed to form brain metastasis. So with that, we were really interested to see whether we can identify molecular mechanism that underline those tropism. So in the same time, so first to understand tropism, we took the approach basically to see whether we can isolate the pure tumor cell from a different organ, and we injected pure tumor cells in several rounds of mice. And if there is a truly tropism, you will see enrichment of certain metastases in certain organs. So in this case, we took the brain metastasis from BRX50, and after 2 rounds of selection, we do see 50% of mice in the 3rd generation can form brain metastasis. So there seems to be enrichment. It's not very 100% pure because we do see increase in the bone and ovary, other organs. So you're probably selecting some tropism, but also you're selecting some cells with general metastatic potential. So this is also, so the tumor also formed much earlier than the 1st generation. And then that's also the case for the lung metastasis. So in BRX7, we took a lung metastasis. In the 2nd generation, you see 100% mice have lung metastasis. And then in this case, in 68, we took a bone metastasis. And then in the 2nd generation, you also see increase of bone metastasis. So it does seem to there is a certain tropism inherited in those cells. And then next we did, in each generation, we also saved certain tumor cells and stromal cells. We did RNA-seq and ATAC-seq, try to understand what are the mechanism for the metastasis. So this is the summary of the, in triangle are the parental cells we injected, and then in circle are the metastatic variants, and the color-coded by each patient. So this is the PCA plot of RNA-seq data and the ATAC-seq data. You can see this is very clearly patient-by-patient driven event. So this is not surprising because each patient have different driving mutations, a different epigenetic landscape. So despite there are differences in the transcription in the metastasis, they still cluster patient by patient. And similarly, epigenetically, the open region, chromatin region also cluster patient by patient. So then we look at, whether we can find a gene signature that specifically that correspond to the brain metastasis, bone metastasis, lung, and ovary metastasis. So we do see some common genes. We also see some unique genes. For example, in the brain metastasis, we see about 700 genes that uniquely upregulated in brain metastasis. And if you look at the gene, pathway analysis, we actually see a lot of gene signature involved in some of those neurological diseases. So specifically why that is, we have to figure out. So because also the case of a brain metastasis predictive case, we hone in a little bit deeper to look at what the mechanism for brain metastasis. And first, we actually did a copy number variation analysis. Initially, we thought this is probably not a genetic event because from the time you injected, by the time the metastasis happened, even though it takes a couple of months, but it's probably not going to induce any genetic changes. To our surprise, actually, when we look at the CTC line BRX50 parental line we injected, and then brain metastasis, pretty much all the chromosome amplification deletion is the same, except chromosome 9q region. We see a very clear amplification in the brain metastasis. And then this is also the case in all the subsequent brain metastasis derived from this first mouse. So you see there's a different way to look at amplification deletion. You see pretty much others are similar to the parental line, And then there's chromosome 9q region amplified in brain metastasis. So we thought maybe this is a selection event then, because the cells we inject are heterogeneous. So to show that, we actually, so this is a collaboration with Jim Hicks in USC. We sorted the single cells that from parental cells injected, and we did a single cell copy number variation analysis. So highlighted here, so this is the parental line in the box. You don't see the amplification. And then in the green box are the 4 brain metastases. You see amplification in the 9q region. And the rest are 61 single cells sorted from the parental lines. You can see that 4 out of 61 cells already carry this chromosome 9q amplification. So it's probably a selection of rare clone that are able to go to the brain and form brain metastasis. So now this gives us an anchor point. So we look at what the genes express in the chromosome 9q, and we overlaid with RNA-seq data we have to look at what are the genes that highly express in brain metastasis compared to other organ metastasis, and also reside in the amplified region. So this leads us to identification of a candidate gene called semaphore 4D. So not only this is fit all those criteria, but also in the data set from John Muskie's paper before, in which they have about 204 breast cancer patients for which they know they did microarray on the primary tumor, and they also know the patient relapse status. We found the semaphore 4D expression in the primary tumor predicted the brain metastasis-free survival, correlated very well, but not much to the lung and the bone metastasis. So then we validated semaphore 4D really expressed high in the brain-seeking CTC lines and the brain metastasis lines. And in another cohort in collaboration with Boudreaux Solier at USC, she have a cohort of PDX models. And then she put the PDX model in the flank of the mice. In this cohort, there is only one mouse that has spontaneous brain metastasis. And this is the one that has highest expression level of semaphore 4D in the primary tumor, also maintained in the brain metastasis. So all those line of information suggests this might be an interesting candidate. So semaphore 4D also is a cell surface receptor. And what's known about, it's mainly known to be involved in angiogenesis and in the exon-axon guidance in the neuroscience field. And what's known about how it works is it can dimerize and bind to the co-receptor, so either plexin B or CD72, and then somehow dimerize plexin B, have this forward signaling. So we want to see what are the co-receptor expression level in the mouse. We found that in the brain tissue, the plexin B1 co-receptor expressed really high in the brain. And then next, we found that which cell type expressed high in the plexin B1 co-receptor. We found in the brain-specific endothelial cells, it expressed very high level of plexin B1. And this is compared to lung endothelial cells, astrocytes, and the parasites. So this gives us a very simple model. We think the CTC is circulating in the blood. Once it gets to the brain region, semaphore 4D could be binding to plexin B1 that expressed in very high in brain endothelial cells, somehow sequestering cells there and help cells cross the blood-brain barrier. So this is the first critical step for the brain metastasis formation. So to test this, we use this in vitro model in which you can culture human primary brain-specific endothelial cells on top of the transwell membrane, and then you can co-culture the parasite on the bottom of the membrane, and you form this proper polarity. And you can co-culture the astrocyte on the bottom of the membrane or on the bottom well. So basically, this is an in vitro way to recapitulate the BBB model. And then you can also use the fluorescence labeled BSA on top to see how much them leak to the bottom to basically to see how tight that is. So with this model, you can put tumor cells on top and measure how many cells can cross the blood-brain barrier. So what we did is we overexpressed semaphore 4D in a different line, BRX7, that has no expression of semaphore 4D and never formed brain metastasis. And then in the BBB assay, so this is control whether no BBB. And then in the BBB assay, you can see that when you have high expression semaphore 4D in every permutation, either 2 cell type or 3 cell type, and that significantly promoted cells transmigrate to the bottom of the well. So this is really support of a hypothesis. So the next question is whether this is really mediated by semaphore 4D binding to plex B1. So what we did in the next experiment is we pre-incubated semaphore 4D overexpression cells with a peptide, plexin B1 peptide that can block the binding of semaphore 4D with plexin B1, and then did a BBB assay. So then we can see that with increased concentration of peptide, you blocked the cells that can cross the blood-brain barrier. in the SEMA40 overexpressing cells, but there's not much change on the basal level of the cells where they don't have SEMA40. And conversely, we also did it the reverse way. We pre-incubated the recombinant SEMA40 in the blood-brain barrier model, and then put a tumor cell on top. So first they saturate the binding, and then the cells cannot bind, and that also significantly suppresses tumor cell transmigration. So this shows that really the cell membrane bonding to the SEMA40 in CTCs bonding to the brain endothelial cell plaques and B1 is really critical for cells to cross the blood-brain barrier. So this is in vitro, and then we did an in vivo model. What we did intracardiac injection in day 3 and day 7. So either control cell or high SEMA40 expression cells. In day 3 and day 7, we isolate the organs, and then we actually lysate the whole organ, and we did a luciferase assay. The reason is because there are only few cells can cross the blood-brain barrier. It's really hard to section exactly the right section. So in this way, we can analyze the whole organ where how many cells can cross. So in this assay, we can see both in day 3, day 7, you see more tumor cells cross the blood-brain barrier when you have SEMA40 expression. And there's not significant difference in the lung, in how many cells in the lung. So this is in vivo showing that SEMA40 does promote cells cross the blood-brain barrier. However, the surprise came if we keep this assay for longer term. So if we keep this for like 3 months, surprisingly, we don't see increase of brain metastasis formation, like overt brain metastasis. So this suggests that it could be SEMA40 can help cells cross, but not sufficient for cells adapt and grow in that environment. But first, SEMA40 is even important at all. So we take a different cell lines that we know is a breast cancer cell line, triple negative line, MDM231 cells. So we know that it has high expression SEMA40, and we know that it can form brain metastasis. We knock down by hairpin, and then in the long-term experiment, we do see decrease of brain metastasis. So this is showing that this seems to be important, but by itself is not sufficient. There must be something missing in the picture. So we went back to our RNA-seq data, where we not only sequenced the tumor cell, but also sequenced the brain metastasis stromal cells. And so when we compare the 3 mice that has brain metastasis, the stromal cell RNA-seq data versus normal stromal cells, we see huge upregulation of genes involved in immune signature. So this is surprising to us because this is an immune deficient mice, NSG mice. And then we look at what are the possibilities. 50% of genes are overlapping with another data set that shows activated microglia. So microglia is resident macrophage and is present in even the immune deficient mice. So we do see that if you directly inject tumor cells in the brain, you do see recruitment of active microglia. And activated microglia has been shown that it can release some innate immune system activities, basically by releasing some of the redox, ROS, and also directly engaging cells. So we want to see, so this updated model is maybe SEMA40 can help cells cross, but then activate microglia. And then you need some other factors coexisting to help cells adapt and grow in that environment. So we went back to our RNA-seq data to look at what other genes highly expressed in brain metastasis compared to other organ metastasis. And the number 5 gene is MYC, and it has highest fold increase in the brain metastasis. And then, so interestingly also, MYC is amplified in the the CTC line can form brain metastasis, but not in the BRX7, the line that we modeled. So there's also another line, BRX68, also has MYC amplification, but it has no SEMA40 expression. So that will come back interesting later. So this is confirmation of MYC expression. So MYC kind of do many things, as we know. And one of the old literature shows that MYC can directly upregulate certain redox enzymes. And that may help cells survive in some certain environment. So we look at we did qPCR to look at the key redox enzymes that are involved in reducing the reactive oxygen species. So you can see that in the brain-seeking lines, we see significant, we see high MYC expression. We also see significant upregulation of the key redox enzymes. And if you directly overextract MYC in BRX7 cells, you do see increase of those redox enzymes. And if you suppress with MYC inhibitor to inhibit MYC and translocation nucleus, you do see suppression of those key redox enzymes. And if you treat those BRCA7 control at the MYC cells with hydrogen peroxide, you see increase of ROS level in the tumor cells, but much less in the MYC overexpression lines. And there's much less apoptosis in the MYC overexpression condition. So the key question, if you co-culture with microglia, what happened? So this is experiment what we did is complicated, so I'm going to describe here. So we did a co-culture, so where you have transmembrane, and then we measure that we'll put the tumor cells with without MYC overexpression on the bottom. And then this is measuring cell intracellular ROS level of the tumor cells on the bottom. So we put, this is control with nothing on top. And this is we treated it with hydrogen peroxide. You see, intracellular ROS level goes up. And this is when we co-cultured the microglia cell line on top of the membrane. And if we measure the ROS level on the bottom, you don't see any change if the microglia are not in touch with tumor cells. In the next condition, we put a tumor cell together co-cultured with microglia on the top, and we measure the ROS level on the bottom. You suddenly start to see increased ROS activity, intracellular ROS in the cells, in the tumor cells. And then this can be mitigated by tumor cell overexpression MYC level. So this is showing that tumor cell has been in contact with microglia to active microglia, and then they can release ROS and then being accumulated in tumor cells. And this is probably one of the limiting factors for tumor cells to grow in the brain. So then we did a series of gain of function and loss function MYC. So in the BRCA7 cells control, we know that it directly injecting into the brain, it will not grow. And if we put a MYC there, you start to see tumor cell growth in the brain. Even though in vitro, when you look at a proliferation, there's no big difference of the KIC7 positive cells compared to control and then MYC overexpression. So we also did a loss of function experiment in BRCA68. And so this is the one that has a high expression and an amplification of MYC in BRCA68. And if you knock down with hairpin, you see that it suppressed the tumor growth. And interestingly, if you look at the tumor growth, in the first 4 weeks, the control and knockdown doesn't seem to have any difference in tumor growth rate. Somehow, suddenly at week 4, 5, you start to see the separation of the tumor growth. So we haven't done the series. So that will be interesting to see whether there's a time that microglia being activated and recruited and then start to have an effect. And having MYC that time will make a difference. So this is, we validated in, so the next question is, if you put Semaphodi and MYC together in the CTC lines, whether that can increase brain metastasis. So we overexpress Semaphodi in the BRCA68 cells that already have MYC amplification. And then we did intracardiac injection, and then we carried this for 3 months. And then we see significant increase of brain metastasis for this condition. And there's not significant difference in other organs, for example, lung and the bone. You don't see a dramatic difference compared to control cells. So, how about this in human? In collaboration with a neurosurgeon, Frank Antinello at USC, we started to collect brain metastasis surgery tissues. And so far we collected 12 of those freshly isolated surgery removed brain metastasis cases. And we did a serial, consecutive session staining. So this is here, we stain cytokeratin to look at where is the tumor cells. And in the next session, we stain for MYC and Semaphodi and also microglia. And here we co-stain it with the tumor cells and the microglia. So in summary here, we see 7 out of 12 patients we found, we do see Semaphodi expression. And in 9 out of 12 patients, we see MYC highly expressed. And in majority case, we see activation of microglia or macrophage, so in the tumor region. And interestingly, in the MYC, if you compare MYC high and low cases, if you look at the KIC7 staining, there's really no big difference in the proliferation rate, at least based on the KIC7 staining. However, if you look at tunnel, there's a big difference. If you have MYC, there's much less apoptotic compared to when you have no MYC. There is a huge separation based on this cohort of data. So we think in this context, MYC could be mainly activate survival mechanism, also promote proliferation, of course, but also, activate, survival mechanism for adaptation of tumor cells in the brain. So now if you go back to the data set where the metastasis-free survival analysis, if you separate by both MYC and SEMA4D, you can see that if the patient have both high expression SEMA4D and MYC, that has significantly worse progressions, metastasis-free survival for the brain metastasis. So in summary, what we found is in this circulating tumors are very heterogeneous, and some rare clones, either by genetic or epigenetic reasons, there are SEMA4D high expression. It exists probably in rare clone in some conditions, and that can bond to the plexin B1 and that express in the brain endothelial cells, and that helps cells cross the blood-brain barrier. But then, many cases, their MYC amplification already exists in the CTCs, and when the tumor cell cross, it will activate microglia, but if MYC exists there, that can help cells adapt and grow in that environment. So two different pathways seems to working on orthogonal mechanism, but together they promote brain metastasis. So in a philosophical thinking, you think this might make sense because brain metastasis typically occurs very late in many of the disease, cancer type, for example, breast cancer, in some melanomas happen late. In the lung cancer, sometimes can happen early. So you probably need accumulation, many of those mechanisms that work together to support brain metastasis. So it's a very inefficient process. So this is the paper. Hopefully we can, come out soon. So basically, I told you this is a brief summary. Patient-derived CTC launch recapitulated metastasis pattern of patient, and there seems to be organ tropism programmed in CTCs. And then in certain cases, we do see clonal event that can drive brain metastasis. So in that sense, not only the CTC numbers matter, but also the quality of CTC matters. So you probably just need one bad CTC to drive brain metastasis. So we need to know which cells we should monitor, which cell we should care, not just like treat all of them equally. And then the last point I have summarized, some of them promote transcription migration and MYC promote colonization of tumor cells in the brain. I don't know the time, actually. Right. So I'll briefly mention two other new developments we have in the lab, because we've been very frustrated with current technologies that can allow us to analyze single live CTC from beginning to the end. So the technology, either you fix the cells, you stain, and you can do a lot of functional and RNA-seq RNA-seq analysis, or you get with impure cells, you have to do further staining and manipulation. So this is two projects. One is to aim to be able to isolate the live CTCs and pick live CTCs, and the other ones, we did a small molecule screen to see what are the better culture conditions for single cell growth. So this is project led by a great postdoc, Mohammed, and this is led by visiting scholar Tang. So in this assay, we actually use a commercial available system called RareSite, and we adapt this. This is RareSite where what they do, they have this tube in the whole blood sample and go to this tube separation. They have this float basically by density. You can separate red blood cells, and then all the Buffy code contain all the nucleated cells. So this is have high capture efficiency of all the nucleated cells. And then you can then collect all the Buffy code, and then you can, in their assay, you can spread on the microscopic slide, and then you can stain whatever marker you want. And then they have this automatic staining, scanning, fluorescence-based system, and they also have automatic picking needle. However, the system was built for the fixed samples. So what we try to do is we try to adapt a live staining technology on this. What we did is we used the first step, basically to get all the nucleated cells. And from there, we have two different approaches. One is positive selection, one is negative selection. In the positive selection, we will rely on cocktail of cancer surface markers, EpCAM, HER2, EGFR combination, live stain on the Buffy code. We'll counter stain with immune cocktail markers that include CD45, CD15, CD16 that will label 100% of the, close to 100% of the immune cells. And then we'll directly put those cells onto the chamber slide and use their scanning system to identify which cell has positive markers but negative for immune cell markers. And this will be able to allow us to pick single cells and then deposit in the PCR tube and do single-cell RNA-seq analysis. On the second arm, we don't want any antibody to stick on the tumor cells because we want to see whether we can culture clonal single CTC to make clones of CTCs. So in this assay, we use immune cell markers, but we use a live cell dye. And we test a few of them, eventually use a cell tracker. So this way we did a similar process, but then we will look for those live cell dye positive, but immune cell marker negative cells. And they will pick those cells and then grow them as single cell clones. So this is an example showing the positive selection that you are able to identify the positive cells based on the EpCAM. And this is, you will find this large live cells, but only positive for the green, but not for the immune markers. So we're able to show that we can spike in single cells, we can retrieve back single cells. And so this have a high efficiency and accuracy. And then this is an example of the picking. So you can find the cells, and this after pick, and this is the field of view, and then you can deposit in the PCR. You can put another well, and then you can pick a pure tumor cells and put another PCR tube. And with this, we were able to do just a proof of principle. We spiked in different CTC cells, and we picked single CTCs from 3 different lines. And then we picked 3 immune cells from the same process. And we performed a single-cell RNA-seq. You can see clearly separation of CTCs versus immune cells. So showing that this is compatible with CTC, single-cell RNA-seq. So in the live cell culture, we were able to show that even though cell tracker does show initial in the first 6 days, it does delay cell growth, but later it does catch up in the proliferation rate. So this negative selection would be compatible for the CTC culture. So this is the technology part. And we're always frustrated with how inefficient for the cells grow. You would think those are really tumor cells. They'll be easy to grow, but it's very, very challenging. So one of the reasons could be they are not in their immune environment. They are suspension. They are epithelial cells, but they are in suspension. And then they probably don't like to be, single cell by itself. So we found that in the CTC lines, even though they grow well as a clump, but if you separate them into 96 wells, the rate of growth from the single cell clones is very, very low. So we use that as a platform to do a small molecule screen. So this is a painful screen manually done by the visiting scholar for 3 years. So what she did is she sorted this BRX68 single cells into 96 wells. In the first day, she will count every single well, make sure they're single cells. And then she will manually add either cytokines or small molecules. And then she will change the media every 3 days. She will manually count every 6 days. Because you cannot just do one well for one molecule because we're looking at the population. So we had to do at least in the first round. So this is total for 4 weeks. And so she did 20 cytokines, about 300 small molecules. In the initial screen, each molecule is 12 to 18 wells. And then we narrowed down a list of molecules, and we did a large screen, further validation with 60 to 100 wells for each molecule, and across 4 CTC lines. So we do have a list of molecules can help cells grow. And interestingly, some molecules have effect on one line, but not on the other. So this is a few molecules. This is one of them showing that it's equally working across 4 different CTC lines. So this is compared to control, and this is adding a small molecule. Over time, each of them have increased the proliferation from single cells. So even though it's not ideal, it's not started from patient, but this is from established lines. So now we can start to use this better condition to see whether we can establish more of CTC lines from patient and then basically increase the power of analysis. So this is what we have so far. So with that said, I want to thank my lab. It's really fantastic. The Metastasis project was led by the really fantastic postdoc Remy Klaus. And then Mohammed Attan worked on other projects. And my collaborators in USC and outside USC, and my funding sources. So thank you very much for your attention. I'll be happy to take questions. Yes.
So I was kind of struck by the end with your single cell, your finding small molecules.
Yeah.
What about what kills them?
Yeah. A lot of kill them.
See
Yeah.
what's they're sensitive to and use that for drug development.
Yeah, that'll be interesting. Yeah, so we actually do find a lot of drugs that do kill it. So a lot of the epigenetic inhibitors, some of the typical kinase inhibitors. We actually found, sometimes we have to figure out whether it's just toxicity or it's actually functioning. So that's an interesting point. We could just test, large scale to validate. We actually found one molecule very interesting, and that keep the cells as single cells across the whole time. And then that never grow and never die for like 24 days. And later, when you take off, they eventually die. So, and then if you apply this molecule in the cells that already grow, expand to 50 cells, you put it there, immediately stop, 50 cells. So that is the molecule we were thinking to follow up. Maybe that's a molecule can initiate, cause cell senescence, maybe less a toxic drug, but more like kind of specific for that context. So we're going to follow that molecule. Yeah, it's an interesting point. Jennifer?
Hi, that was great. I was wondering if, similar to the SIM7 or SIM4A.
Yeah. SIM40, yes.
And then the receptor. So the receptor is sort of specific for endothelial cells, particularly in the brain.
Not really, actually. So other brain cells also express plexin B1. For example, SIM40 plexin B interaction has been long studied in neuroscience, and then particularly in Parkinson's disease, Alzheimer's disease. So I think, microglia, astrocyte could also express plexin B1. So we didn't look at whether SIM40 can activate microglia in that context. We didn't do that yet.
What I'm wondering is how much of the other organ tropism could you potentially look for other receptors that were specific to endothelial cells in those particular organs?
Yeah. So, I think that single cell data started coming out from, different endothelial cells at a different organ. So we haven't, specifically look, maybe we can find some specific receptors that, specific for a certain organ. The ligand. Yes. Yes. Yeah, we haven't done that. But I think, there are single cell data start coming up from the endothelial cell part. Yeah, that'll be interesting. Yeah. Hi, Tracy. Thanks. Yeah, good question. So because SIM40 has been implicated in angiogenesis, we haven't done specifically looking at difference, but when we first found out the disappointing result, when we did 3 months, we thought, okay, what if we directly inject cells into the brain? Maybe there's angiogenesis. We directly injected in the control cells and SIM40 overexpression cells into the brain. We still didn't see increased angiogenesis. We didn't even see increase of brain metastasis at all. So I think that's the only thing we have done. We haven't looked at, in the PDX whether there are more, blood vasculature, with CD31 staining. We haven't done that. That's a good point. Yeah. Hi.
I'm wondering about your mix results.
Yeah.
The argument is that mix is protecting the cells from ROS.
Yeah.
Allowing them to survive.
Yeah.
And I'm wondering if you think that is the main contributor, but you intentionally did this with you.
Yeah, I didn't show the.
Any of the genes that were operated that.
Yeah. That's a good point.
Or whether it could be also, for example, mediating sort of stem characteristics to making cells more plastic and therefore making them able to survive.
Yeah.
Do you know that?
So, that's the last part data I didn't show. I told you about. So it's for the revision. I didn't get time to show. So what we did later, the reviewer asked us to see how general that effect is. So we only analyzed like a few lines. So then we overexpressed MYC in the MCF7 T47D cells, and then we inject into the brain, and then that hugely increased the brain metastasis formation. And then we look at the downstream of those redox enzymes. They're included GCLC, GPX1, and 3SD SOD genes. And even the CTC line seems to all high. But if you look at what are the genes consistently up, it's GPX1. It's consistently up in all the cell lines that we overexpress MYC. And in fact, in T47D, that's the only gene that's being up by MYC. And so what we did is we knocked down GPX1 in the MYC expression T47D cells and injected in the brain. That rescued the effect. that suppressed the MYC promoted growth. So I didn't have time to put it there. And at least it's showing that that's important through GPX1, but we don't know whether that's the only reason. Yeah. So it could be other effects also exist. Yeah. Wait. Okay.
Civic has been shown to be a strong immune suppression user.
Yeah.
Why do you think Civic specifically increased the brain involvement of immune system?
Yeah, so we don't know the mechanism. So it could be, in certain contexts, MYC has also been shown that it's general as a transcription amplifier to set up the epigenetic state. So maybe under stress condition, there are some readout here, MYC somehow help amplify certain genes, help cells survive. So we don't know why specifically in the brain that's the case. And, there are papers showing MYC can directly upregulate PD-L1, CD47, but it has to be very high level. We did look at the PD-L1, PD-1, and CD47. We didn't see a very clear trend, at least with our overexpression. So we didn't go down that route to validate. Yeah. There was a question. Yes.
I remember one of the tables showed that the person made 4 lines.
Mm-hmm. Yeah. Yeah.
I wonder, now thinking under the umbrella of maybe circulating cancer cells are just a very small fraction of the time, that discrepancy simply because the obedience of the cancer is not enough. In other words, there
could be. Yeah.
number of ingredients of already are circulation mutation, and they actually have more tendency to become CDCs.
Oh, I see. More tendency becomes. Yeah, that's an interesting point. Yeah. So I think now that's if it's not treatment induced, or even treatment induced, I think the common notion, select. Yeah, there are several papers saying that if you sequence deep enough, you could start picking up a very low allele frequency in the primary tumor, some of the rare mutation that later got increased. So I think that's definitely showed that's the case. Yeah. That could be interesting to test, but I don't think anybody has tested looking at the same model, sequence the primary tumor deep and then see whether CTCs, that's the reason more CTCs happening. So we don't know. Yeah, that's a good point. Yeah, could be. Yeah. Mm-hmm. Yeah, that's what we plan to do. Yeah. Either pre-treatment options. Oh, okay. There is a company made antibody specifically inhibit the binding of plexin B1 and SEMA40. This company initially developed this therapeutic for Parkinson's disease, but later they found SEMA40 involved in angiogenesis. They actually started to do clinical trials in the patient, but not particularly for brain metastasis context, but just for cancer in general. So that could be interesting, but I don't know. So the question would be the timing will be tough because you don't know when and how long you should treat the patient. There are also studies showing that CT could shed pretty early, whether when you start treatment, it's already late. So one thing I thought maybe will be interesting to test to see is to predict using SEMA40 MYC as markers. You can predict which patient, for example, lung cancer patient, 50% have brain metastasis. Nowadays, people, they are recommended to do a whole brain radiation therapy as a prophylactic. If we can predict which patient might have high chance of brain metastasis, maybe you can use that to stratify patient for prevention. The other thing we're thinking to look at what exactly mechanism how SEMA40 mediated trans-BB migration. Initially, we thought maybe the binding can create a gap, cause the leakiness of blood-brain barrier, but we found exactly opposite. When we did recombinant SEMA40, that stopped tumor cell cross. That means it's not just creating gaps for let tumor cell cross. So we think it has intrinsic properties in the tumor cells that mediated by SEMA40. We think it is reverse signaling that we're going to try to look. Yeah. All right, great question. Sorry, do you have a question?
I guess one last question is, here, the mice, when they get disease, does that actually kill them? And I was actually kind of wondering also, outside of luciferase manipulation,
Yeah.
you actually look them by PET-CT and see if
Oh, yeah. Yeah, we didn't do the PET-CT, any of those. But yeah, in some of the mouse, we have to sec them at the end. You know, we got notice that they do get sick, so we have to sec them. Yeah. Oh, we didn't let them die because we didn't do a survival one. So the initial, we were just thinking we see steady metastasis growth, and then also sometimes we got notice that mice are not doing well, so we had to sec the mice. Yeah. All right. Thank you so much. It was a great talk.